Okay, so more evidence of how things are related is by biogeography. This is the study of plants and animals, how they're distributed around the world. So remember that at one time all of the continents were together. What was that called? Pangea. Okay, and because of Pangea, okay, we have animals that are found on 2 different continents because they crossed over, because they were able to back then. So if you look at South America and Africa, you can see how things crossed over there. Some things went all over the place. This is a plant, a fern. It's found in pretty much all of the continents, even Antarctica. So that's a way to study how things spread out over time, how they ended up where they ended up, what things are related to what. Okay? So this is a way to observe migration patterns and how things changed once the continents separated from each other. Did everybody copy this already? Okay. All right, we're going to come back to that. All right, and then the last thing is called sequence homology. Okay, so looking at fossils and looking at migration patterns and stuff, that's nice. But we have technology now where we don't actually need that stuff anymore. We can look at DNA to determine relationships, also RNA, and also protein sequences. So in the past, if you've had 2 animals and you're trying to figure out if they're related, you just kind of look at them, see if they look similar, and put them in a group together. Now we can do DNA testing, we can do RNA testing, and we can do protein sequence testing. And if they have similar sequences, it's called sequence homology. There's that word again, same sequences. So the more similar they are in these sequences, the more closely related the species is going to be. So if you look at, this is humans compared to chimpanzees, we're 99.6% DNA is the same. If you look at our protein sequence, however, we're 100% the same for chimpanzees. Fruit fly, we are 72.4% similar to a fruit fly, and pretty similar there with the proteins. A worm, 68.2% similar DNA to a worm. Okay?
What does that mean exactly?
That means if you take a DNA sample from us and a DNA sample from a worm and you scan it like a barcode, 68% is going to be the same.
And
exactly
why do we look so different?
Because of the part that's not the same.
So there's a
That's a short answer.
So there's a 68.2% chance that the DNA of a human could be mixed up with the DNA of a worm.
No.
Don't know how people do the worm like on dances?
Oh my God.
There you go, that's how they were.
That indicates our close relationship with worms. Yes.
I'm assuming this has
All those worms I enjoy.
2% sure that is not right.
Right. So you're also going to need to be able to look at these. These are called electrophoresis gels for DNA. So here, this is comparing 4 deer species. Here's a common ancestor to the deer species. And the way you read these, similar to a barcode, is this is their DNA, and this is the part that's similar. So if it lines up, like this part of their DNA is similar, this part is similar, this part's similar, this part. So number 3, it looks to be very closely related to this common ancestor, probably the most closely related. 2, not so much because it doesn't have as many similarities. So that's a way to tell not only that they are related, but how close they're related they are. All right, so we know DNA is a molecule with our genetic instructions. The instructions are to make proteins. So a lot of times that's why your DNA similarities and your protein similarities are going to line up. So DNA sequencing technology makes it easier to compare these sequences. So this is what she was asking about earlier. So you have species A and species B. Remember, DNA is just a sequence of these 4 letters, A's, G's, T's, and C's. And if you just line them up, you can look for differences. The more differences, the less closely related you are.
Oh my gosh, we're almost done.
We are.
Yay.
So questions on that? We pretty much answered that already. All right, so in addition to DNA, you can also dive further into sequences. You can do amino acid sequences, which is going to determine protein sequences. There is a protein that most living organisms produce, which is called cytochrome C, which is involved in the production of ATP. So everything has to make energy, so everything is going to need ATP, so everything produces a cytochrome C. So you can do a sequence for cytochrome C and also see how closely related things are. It's another thing you can look at. Okay, and for each protein, you're going to count the number of similarities in amino acids and then calculate a percentage of similarities. So the higher percentage, the more related the species are going to be. So there's just a little math equation here. So number of amino acids that are the same, and then the total number of amino acids is going to be your percent of similarity.
I'm glad I'm never gonna have to do that.
Yeah, computers are going to do it mostly. Like, you don't have somebody sitting there counting all those H's, T's, and C's. That would be a terrible job. All right, so here's cytochrome C, and you might see a diagram similar to this on your test or on the state test. So these are abbreviations for different amino acids that can be found in proteins. Here's a human. Okay, so percentage of amino acid sequence similarity with a human. So obviously a human and a human are going to be 100%. A horse is 88%, whale is 90, penguin 87. So a lot of them are very close, even down to here's a reptile, here's a bug. Okay, bugs are least related of animals. And then rice, we're 60% related to rice. Okay, so plants are living things too. They have to make energy. They produce cytochrome C just like everybody else. I think that's it. Is that it?
I forget how we're related to a traffic turtle. Some of y'all mean.